Thanks. Hi, everyone. Thanks for the introduction. So I think my talk is going to sound a bit like team 2 compared to the talk before. So I'm not really, oops, this is not supposed to. Looking at connectivity, but rather this localization. So how is information, functional information organized in the cortex? And some of my main background and my main interest is in vision. Which means try and understand how visual information is organized and processed, and how can we actually make sense of the world surrounding us. And we can look at this at different levels. So you can imagine having a scene, and we can perceive that in terms of low-level visual features, so things like orientation and contrast, or a higher level, so faces, objects, and somewhere in between. And the other aspect of a visual scientist is we love illusions. So hopefully, if I show you something like this, you are desperately trying to make sense of all this cacophony of lines. And maybe at some point you will start and see some shapes in here. Anyone can see any specific shapes being repeated at all? No? Maybe if I give you a bit of a hint. Oops. No. Okay. Those were circles, and now probably you can't stop seeing circles everywhere. And there are also some rectangles in there. There you are. So what I'm trying to show here is once you see it, you can't see it. And there are these mid-level, what we call mid-level visual features. There are contours that basically allow us to extract objects from a background. And it turns out that curvature or edges and contours can be processed and are organized in a specific region of the brain. And the region that we're talking about is the area V4, which is located in the ventral portion of the occipital lobe, as shown by that purple circles up there. And it's a very interesting region and location for the human brain in terms of vasculature, because we have the transverse sinus that is passing exactly at that level, which complicates a lot when we measure with fMRI. However, part of the sorry, I just noticed. Sorry, that's a bit complicated because it's not showing the actual. Okay, let me try and explain it. So part of the study was done with electrophysiology in monkeys, and what happened was they basically measure responses to curved stimuli versus straight lines. And what they observed was a map, effectively, that were blobs that showed preferred responses to curved lines versus other blobs that were showing preferred responses to straight lines. And those were organized in a very specific pattern. So you can imagine like a stripe patterns of preferences for curvature, preferences for straight lines, curvature, straight lines, straight, and so on, over the span of area V4 in macaques. In human, however, this type of fine resolution has not been able to be achieved with standard fMRI. However, using bold fMRI, it has been shown that there are patches along the ventral visual stream that are selective for curvature. So when we present, for example, balls versus squares, these areas have a preferential response for the rounded shapes compared to the squared ones. So part of my project here was to try and use a higher resolution and try to dig deep into some of these areas, specifically before, and see if we can identify these domains, so these stripy patterns or columnar organization for curvature in humans. And how did we do that? So first of all, we had to identify where V4 is in human brains. So on the left-hand side, you have a representation on a flat surface of the visual field, of a visual field map. To obtain these, we basically use a checkerboard stimulus that is a wedge that is rotating at a specific frequency and phase around the screen and stimulating specific timings. And by doing that, we can decode the time at which we are stimulating and the location in the visual field and map that back onto the visual cortex and use that information to identify different brain regions based on the borders that they are encoding. So from V1, V2, V3, and V4. And basically, once we have the V4 response, the V4 area, we are then looking into that specific region. Is there a preference response for when our stimuli are straight lines or concentric squares versus when our stimuli are concentric circles? So that is the kind of response that we are expecting. If the response is preferential for circles, I should see an increase in response in this case versus that case. The actual design was a simple block design. So we have a 30-second stimulation where we are showing these concentric circles, then a 30 seconds of rest, and then another 30 seconds of concentric squares, and so on for 15 minutes roughly, and then repeat that up to an hour. The important thing to avoid that people would get adapted to the stimuli is that we changed the contrast at every half a second of the stimuli. So here you can see that we start from white at the center, black, white, black, white, and so forth. The next presentation would be the opposite. And interestingly, this gives a kind of induces a motion effect. So participants get kind of hypnotized during the scan, which either makes them fall asleep or be very awake. The next thing that we wanted to understand is obviously our stimuli differ mostly, along the horizontal and vertical meridian. So if I assume that this is the portion of my visual field that V4 is encoding, then my stimuli will differ the most along the horizontal and the vertical meridian, as shown here. So I would expect that response if I map exactly these portions of the visual field. Whereas if I look at the obliques, even these concentric squares are somewhat rounded around the edges. So all that changes is a kind of gradients of curvature. So I have less curvature information in the concentric square, but there's still curvature when compared to the circles. So my response should still be roughly the same here. Okay, so as I said, the previous studies using fMRI, they used standard bold fMRI, which we know it's an indirect measure of neural activity. And that is because we rely on neurovascular coupling. So what I'm trying to show here is in our voxels, we have we include very different structures, and we know that when there is activation, all that happens is there is an increase in energy demand at the postsynaptic sites, which then triggers an increase in blood flow, an increase in blood flow, an increase in blood volume, and then there is some oxygenation exchange even at the venous side. And most of the times this happens even at a different location from where we're actually measuring, which obviously is a problem where we're trying to identify very specific information in space. So to try and make up for this problem, we decided to use vascular space occupancy, or VASO. And I have to highlight the fact that this study was done at 3 Tesla in York. So the actual sequence was provided, developed and implemented by Renzo Huber and Rudi Sternberg. And the reason why we decided to use vascular space occupancy is that, first of all, it provides both bold images. However, we know that bold signal is biased toward the surface. So where if we think about the thickness of our cortex, where we have white matter down here, and the main vasculature is located at the surface. So most of our signal in bold is weighted towards these areas, whereas VASO is weighted more toward the microvasculature. So we will get deeper layer informations. Again, because we are trying to understand how information is encoded in a columnar organization, we want to have that spatial specificity. So how does VASO actually work? Basically, we start with a 180 degree inversion pulse, and we rely on the fact that gray matter and blood have different T1 timings, which means we start with this inversion and then we let the magnetization relax back. The gray matter will relax at a faster rate compared to the blood. When the blood signal hits the 0 line, we then excite. such that we null out the blood information and always measure is the measurement from the gray matter from the tissue. So another way of thinking about this is if we imagine that our typical voxel is made up of 94% of tissue and 6% of blood, we still get most information from our tissue, right? So at the time of inversion, everything is, sorry about that. We're at this stage, then during baseline, where nothing is happening. So let's say during the rest condition, we have a specific proportion of tissue. When there is activity, we have vasodilation. So we have the vessels that are enlarging to increase the volume, blood volume increase, which means we have a decrease in tissue. And that's what we're measuring. So basically, it's a decrease in the MR signal. Excuse me. And that is the to the rescue. Thank you. So we have a decrease in the MR signal, and that is proportional to the cerebral blood volume change. Okay, so what do we actually get when we look at the signal? As I mentioned, this was done at 3T. Our TR was of 2.6 seconds per pair of images. So we get a vaso or a blood node image and a walled image. The resolution on which we collect this data was 1.2 millimeter isotropic, and this is basically just showing the 20 slices that we acquired, just flipping between the vaso and walled acquisition. And when we look at the TSNR map, we can see that obviously walled has a much higher signal contrast, especially around the temporal lobes. But when we look at vaso as well, in the visual cortex, this is still high enough to get good results. So what do we actually get? Let's start with the complicated results first. So here we have each row is a different participant. In the first column, we have just the representations of the portion of the visual field that area V4 for this specific participants covers. You can see that it's quite biased toward the lower visual field compared to the whole of it. Then the second column shows the one-to-one mapping of which parts of the V4 is preferring. I'm really sorry. Is preferring curvature to straight lines. And the same is in the third column is the results for the vaso data. So you remember that I said my stimuli are maximally different along the horizontal and the vertical meridians. And this is what I'm trying to show here. So in bold, for example, you have the maximum preference for curvature along the horizontal meridian in this case, not so much in vaso, but if we bin, this is shown more along the vertical meridian. In the second subject, instead, you see that the V4 representation is slightly different. But again, we have the vaso generally has a higher proportion of voxels, let's say, that are preferential, show preference for curvature compared to straight lines. Now, if we want to just generate a map and actually look at these curvature domains, this is what we do. So we have, let's say we encode a preferential response for circle with red, a preferential response for squares with blue. And all we do is we just slice through our acquisition, and you should start seeing around area V4, which is around here, these blobs of red and blue that start to appear. And if we then want to visualize it in a different way, we can zoom in, so cut out a disk around the area V4, flatten it out as if it was a cake. So we have now a 6 millimeter diameter. And then we simply project the same map that I was showing in this video onto this disk so that we can look at it in a more like cake way and look not only at the surface, but also through depth. And you can see, again, red is curvature, blue is straight lines, and there are some stripes and that blobs go through the depth as well. So it looks like there is some form of curvature domains in human V4 as well. Another way of visualizing it only on the surface is through this visual field maps organization. So again, this is different hemispheres of the same participants. We are focusing on area V4 here, and when we project only on the surface, in this case, so it's you can average it across the depth. You can see that there are these stripes of blue and red across V4. So there is this modular organization of curvature, and this is generally stronger or more defined for vaso compared to the board results. The next slide should show this is some of the participants. So we have again left and right hemisphere for the different participants that we tested. The difference being that different participants did a different amount of testing, but in general we can still identify some level, even for this participant that, for example, did only one run. There is still some level of modular organization between the conditions. And in general, this is much more cleaner for the bold than, this is much more cleaner in the vaso than bold data, suggesting that vaso has a higher specificity. And I think that's all I had to do. I'm really sorry about some of the pictures didn't show. But yes, so just to summarize, we use basically vaso at 3 Tesla to identify for the first time curvature domains in humans before. And if you have any questions, happy to try and answer.